Good morning, students. How are you all? Good? Okay. I hope every one of you are preparing for your first unit test. Yes, I hope so. Children, in our previous video, we have started with our revision of Science 2, right? Today, children, we will continue with our revision number 2 of Science 2. So, let's begin. Revision number 2 of Science 2. Question number 1. Write about the importance of fossils. Answer. Importance of fossil. First, they inform us about the types of living things that existed in the past. They inform us about the extent to which living things have changed over time. Third, the most recent fossil is found in a rock nearest to the Earth's surface. Therefore, they inform us about the time when a particular life form existed. So, basically, this answer tells us about the importance of fossils. They tell us which life existed in the past. Okay, so this is all the importance of fossil. This may come in your subjective paper, which carries 2 marks. Understood, children? Next is write a short note on Lamarckism. Answer. Lamarck gave the principle of use or disuse of organs. He proposed that the reason behind evolution was the change in morphological activities of the organism. Now, for example, children, giraffe. It has a long neck. We know this because it used to browse on leaves of tall plants by extending their neck for several generations. Okay, so by extending its neck for several generations, now giraffe has a long neck because it used to browse on leaves for tall plants. Shoulder of ironsmith are strong due to repetitive hammering movements. Wings of ostrich and emu are weak because they are not used for flying. Legs of birds of swan are used for swimming because they live in water. So these characteristics are termed as acquired characters which are transferred from one generation to another. Understood the answer, children? Good. Third, we have define heredity, explain the mechanism of hereditary changes. Answer. Heredity refers to the passing of traits from parents to offspring, either through asexual or sexual reproduction. Gregor Johann Mendel was the first to carry out the study of the transmission of characteristics from parents to offspring. He proposed that heredity is controlled by factors which are now believed to be segments of chromosomes or gene. These traits are passed on in the form of genetic information from parents to offspring. This genetic information is stored in the molecules called DNA. During the process of reproduction, the child receives genetic information from both the parents in the form of DNA. 50% genetic information is passed on from the paternal DNA, whereas 50% of information is passed on by maternal DNA. This is why a child closely resembles its parents and has traits similar to both of them. Commonly observed heritable features are curly hair, a particular type of ear lobe, hairs on ears, etc. So, children, this answer tells us about the heredity and the mechanism of hereditary changes. Johannes Gregor Johann Mendel was the scientist and he studied the transmission of characteristics from parents to offspring. So, children, the genes which are being transferred from parents to offspring it is stored in the molecules, that is DNA, and through this process of reproduction a child receives 50% genetic information from father and 50% from the mother. So that is why a child closely resembles to its parent. For example, we can say curly hairs, a particular type of earlobe, hairs on the ears, etc. These are the some of the commonly observed heritable features. Okay, got it? Let's see the next process, next answer. Explain the process of formation of complex protein. Answer. The synthesis of proteins occur according to central dogma. The central dogma explains how genetic information flows from DNA to RNA to make a functional protein. It suggests that DNA contains all the information required to synthesize a protein and the RNA acts as a messenger which carries this information to ribosome. The central dogma is represented as DNA gives mRNA in the process of transcription and then while translation we get protein. So let's see the first, transcription. Transcription results in the formation of an mRNA using DNA as a template. This process takes place in the nucleus. You must be aware that DNA is made up of 2 strands, isn't it, children? But in this process, only one strand of DNA is required, and this one strand is used as the template for the synthesis of mRNA. Got it, children? An enzyme, RNA polymerase, helps in this process. It helps in copying the information from DNA strand to mRNA. Once this process is complete, the RNA polymerase dissociates from the strand and the newly synthesized mRNA is released from the nucleus. Like DNA, RNA is also made up of nucleotide, but there is a difference. RNA contains the nucleotide uracil instead of thymine, which is present in DNA. This uracil pair with adenine just like thymine. Once the process of transcription is complete, the newly synthesized mRNA is released from the nucleus to the cytoplasm. Now the next process is translation. The newly synthesized mRNA contains information in the form of codon. A codon is a sequence of 3 DNA or RNA nucleotide that corresponds with a specific amino acid. Since these codons contain 3 nucleotides, they are called as triplet codon. Let's see the process now. After the mRNA is released into the cytoplasm, it reaches ribosome where the process of translation occurs. Each mRNA can be made up of thousands of triplet codon. Two other types of RNAs, that is tRNA and rRNA, also participate in this process. Each tRNA has an anticodon which has the sequence complementary to the codon on mRNA. These tRNAs supply the amino acids as per the codons on the mRNA. The amino acids which are brought by the tRNA are joined together by peptide bonds. This process is carried out with the help of rRNA. During this process, the ribosomes move from one end to the mRNA to the other by the process known as translocation. At the end of this process, a protein is formed which undergoes further modification to form a fully functional protein. So children, this is a little long answer, but you have to learn this answer so that it may come in your exam, so you are able to write it. Okay? Let's see the next. With the help of suitable diagram, explain in detail mitosis. So let us study what is mitosis. Mitosis is the process of equational cell division where daughter cells so produced contain an equal number of chromosomes as that in the parent cell. Now let's see the various stages of mitosis in an animal cell. The first stage in mitosis is prophase. It is the first stage of mitosis that is marked by the initiation of the condensation of chromosomal material. Each chromosome is composed of two chromatids, right, which are attached by the centromere. At the end of the prophase, the mitotic spindle begins to form from the centriole. The nuclear membrane and the nucleolus disappear completely by the end of this stage. So what is happening in prophase? There is condensation of the chromatin, right? Chromosomal material. When each chromosome, it is composed of two chromatids and they are attached by a centromere. And children, spindle begins to form from the centriole, and nuclear membrane and the nucleolus gets disappeared completely by the end of this stage. You need to write this description and also you need to draw this diagram. Okay? The next stage is metaphase. It is the second stage of mitosis in which the condensation of the chromosomal material and the spindle formation gets complete. The spindle fibers get attached to the chitinocores of the chromosomes and chromosomes get aligned along the metaphase plate in the middle of the nucleus. So what is happening here? The condensation of the chromosome material takes place and there is a formation of spindle fibers. Then these spindle fibers, they get attached and then all the chromosomes, they are aligned along the equatorial plate in the middle of the nucleus. Okay? So this diagram you need to draw. Third stage is anaphase. It is the stage of mitosis where the centromere of the chromosomes split and the chromatids get separated. So what is happening here? The centromere, okay, it gets splitted and chromatids are separated. And then the contraction of spindle fibers moves the sister chromatids apart towards the two opposite poles. So, these spindle fiber, they move the chromatids apart towards the opposite poles. So, this happens in the stage of anaphase. You need to write and then you need to draw this diagram also. The last stage is telophase. It is the last stage of mitosis. In this stage, chromosomes finally reach their respective poles. The spindle fibers disappear and the nuclear envelope reappears around the chromosome cluster. Also, nucleolus, Golgi complex, endoplasmic reticulum and other cell organelles reappear. Okay? Understood, children? What is happening in the last stage? The spindle fibers, they get disappeared and the nuclear envelope reappears. Okay? And also the various cell organelles like nucleolus, Golgi complex, endoplasmic reticulum and other cell organelles, they reappear. And two daughter cells are formed, two daughter nuclei. You can see here. So this is the diagram which you need to draw and you need to write the description also. So this is the all four stages of mitosis. Okay, what are those four stages, children? Prophase, metaphase, anaphase and telophase. Correct. So children, you have to write all these details. Okay, if this answer comes in your exam, this may come for 4 marks. Okay, so it is an important answer. I hope every one of you have understood this. Good. With this, children, we have come to an end of today's revision. I hope every one of you must have understood the revision. Children, study hard and keep calm. Okay? Best of luck for your exam. Thank you, children.